All right, good morning, everyone. Welcome. Welcome to Thursday. We get to talk about metabolism some more, and a reminder that on Tuesday, I asked you to please complete and bring your metabolism worksheet. So you're going to need that for some of the call and response and activities we do today. All right. So what we covered on Tuesday is just sort of this first line item. We looked at enzymes and different things that affect their function. We looked at different ways of getting energy and carbon. And we looked at the important role of and movement of electrons. So today we're going to focus on catabolism. So aerobic respiration, anaerobic respiration, fermentation. We're going to compare these processes in prokaryotes and eukaryotes. And then we're going to move on to autotrophic processes, ones that are anabolic, building rather than breaking down. Most importantly, carbon fixation, which is a process by which carbon dioxide is taken out of the air and made into sugar by some organisms, and that sort of feeds the rest of the ecosystem. And the two processes we'll look at that do this: photosynthesis and chemolithoautotrophy. Now, if you really want to geek out on microbiology, there's way more to this process than we are going to talk about. We're going to talk about a very small subset of reactions that fall under this category. And we're going to focus on the one that does carbon fixation. But it is just to let you know that it's a bigger world, and there's hemolithoautotrophs that do all kinds of crazy fun things. We want to make sure that as we're looking at all this stuff, we're kind of understanding the practical applications that you'll need in this course. So you want to remember how to do a thioglycolate and why that matters, catalase and oxidase. Enzyme testing is part of our lab this week, and growth in various environments. It is important to compare aerobic respiration, which you've probably studied quite a bit, to anaerobic metabolism, of which there are several different forms. And we're going to touch on carbohydrate testing in this week's lab, in particular the phenol red fermentation. I'm going to just mention OF carbohydrate test like for 1 second in lab meeting. And also, one of the things that perhaps you haven't thought about before is that a lot of microbes, single-celled microbes like bacteria, for example, need to make what's called exoenzymes. These are enzymes that they make, but they send them outside. And it's because, unlike us, they don't have a digestive system. So they kind of have to digest things outside of themselves. So those exoenzymes are sort of like they're throwing up their digestive enzymes outside the cell. So we'll talk about that just a smidge. Okay, so I'd like you to reference, you don't have to tell me, you don't have to submit anything, but I want you to pull out your metabolism worksheet because I asked you to do a drawing on question 2 of a eukaryotic cell performing respiration. And so you need this in order to answer the questions that we're about to talk about. So let's start with this question in the chat. Where in the eukaryotic cell is glycolysis happening? So this is a eukaryotic cell that's performing aerobic cellular respiration. Where is the glycolysis pathway, which is the first thing that happens, glucose being split down to pyruvate? Where is that happening? Great. That's happening in the cytoplasm. Good job. The next thing that happens is that pyruvate, which is the product of glycolysis, is converted to acetate in basically a single enzymatic reaction where one of the carbons is taken off of pyruvate and thrown away as carbon dioxide, and some of the electrons are taken off of pyruvate, so it's oxidized. From a 3-carbon molecule, it's oxidized down to a 2-carbon molecule. And where does this intermediate, so-called intermediate step, occur? It's the step right before the Krebs cycle. Great. Yes. So this process happens in the mitochondrial matrix. And what that means is that in a eukaryotic cell, at the end of glycolysis, when you've made pyruvate, You then have to import pyruvate into the mitochondrion before you can convert it to acetate. All of this is so that you can get this acetate, which you add coenzyme A to and call it acetyl-CoA, so that you can feed it into the Krebs cycle. Where is the Krebs cycle taking place? It's this multi-step enzymatic process that's called a cycle because it kind of goes in a circle. Right. So we start in the cytoplasm, we do our glycolysis, we produce pyruvate, we take that pyruvate, we transport it across two membranes into the mitochondrion interior, which we call the mitochondrial matrix. We convert it to acetate, and then we feed it into the Krebs cycle also in the mitochondrial matrix. Good job. Now, one of the jobs of all these processes together, and it happens in glycolysis, it happens in this intermediate step, and it especially happens in the Krebs cycle, is to remove electrons initially from glucose and later from pyruvate and later from the intermediates in the Krebs cycle. Remove electrons, oxidize those organic chemicals and put them onto NAD+, which creates NADH. So we're taking a whole bunch of electrons out of basically glucose, or what was glucose before it got chopped into bits. We take those electrons and we feed them to the electron transport chain. Where is the electron transport chain located? It's in a membrane, because when you do electron transfers, you've got to be in a membrane. The transfer is kind of tricky. It's not something that you can handle really very easily if you're floating around in a fluid. You have to be in a membrane to kind of stabilize everybody. So it's happening exactly in the inner mitochondrial membrane, which is the one that lines the matrix. Okay. So of course, what happens then is that electrons flow through the electron transport chain, and as they do so, they release energy, because electrons are mostly energy. So we just squeeze the energy out of them, and we use that energy to pump protons, and that proton gradient is used to make ATP. Now, obviously then, ATP is generated in the electron transport chain. Is it also generated in either of these pathways, glycolysis or the Krebs cycle? Great. Yes. So glycolysis produces 2 ATP. The Krebs cycle produces several ATP, and the electron transport chain produces like 32, all the rest. Now, as I mentioned earlier, the big picture purpose of this process is to take glucose, which is a high energy organic molecule with lots of high energy electrons in it, and take those electrons out. And some of the time, in order to do that, you've got to chop the glucose into bits. But really, the point is to take the electrons out. So when you do that, you remove those electrons and you put them onto NAD+, remember our empty electron taxi cab, and that produces NADH. NADH is just what we call the NAD+ when it's full of electrons. When are the NADHs getting made? When is NADH produced? In which of these pathways? Right. Okay, so glycolysis, it happens, when glycolysis happens, some electrons are taken out, and that's when NADH is produced. There's also some NADH produced here during the pyruvate oxidation step. There's quite a lot of NADH and FADH2, which is a similar molecule. I think of it as like a different cab company, is removed in the Krebs cycle. So NADH is produced in glycolysis, the intermediate step, and the Krebs cycle. Now, NADH is subsequently oxidized back to NAD+, meaning those electrons that we put into it now get taken off. Where and when does this happen? Yes. Okay. So it happens at the very start of the electron transport chain. So the very first guy in the chain is called a chain because there's several different components that go in order. So the first guy that accepts electrons from NADH oxidizes it back to NAD+. And that chain is located on the in the inner mitochondrial membrane, and the electron transfer is happening officially on the matrix side of the inner mitochondrial membrane. Now remember, as I said earlier, the point of doing this is to feed electrons through the electron transport chain. And as that happens, as the electrons flow through the chain, they release energy. And the components of the electron transport chain harness that energy to pump protons across the membrane. So hydrogen ions, protons are hydrogen ions, end up accumulating on one side of that membrane, like, sort of water building up on one side of a dam. Where do those hydrogen ions accumulate to form that gradient? Right, in the space in between the two membranes of the mitochondria, what we call the intermembrane space. Now, what happens next is all those protons, which are kind of built up in this space, and they're really concentrated now, they're allowed to flow back through ATP synthase to create ATP. So where is ATP synthase located? Okay, I'll give you a hint. It's located in the same place as the electron transport chain. And some of you have gotten there, but I did see a number of question marks, which I appreciate. Great. So it is in the inner mitochondrial membrane. Now, per glucose in a eukaryotic cell doing aerobic cellular respiration, how much ATP is made per glucose? Great. And it is a little bit of a rough estimate because this process is not 100% efficient every time. And so if you read different textbooks and so on, you'll get sometimes different numbers. We're going to go with the most commonly accepted number, which is 36. Okay. Now, all of that was review from previous courses where you should have taken this already. So this should be review. You should kind of just know this stuff. And if you didn't have easy access to it, then you need to just dust off that part of your brain that does hold this information. What we're going to focus on now, though, is what you might not have covered before, which is where is all this stuff happening in a prokaryotic cell? Because we have, hopefully by now, I've bashed you over the head with the idea that bacteria can do aerobic cellular respiration, even though they don't have mitochondria. Not all of them do, but they are capable if they have the right enzymes. But they don't have mitochondria, so there's a lot of stuff on this chart that wouldn't make sense. So where is all this stuff happening? Glycolysis, pyruvate oxidation, and the Krebs cycle all happen in the same place. Where is that? Great. Exactly. It happens in the cytoplasm. And so interestingly, what that means is that when you do glycolysis and produce your pyruvate, you don't have to transport it across two membranes to get into the matrix before you do stuff with it. You get to just use it right away. Okay, the next part is the really fun part. So in a eukaryotic cell, the electron transport chain and ATP synthase are both located in the inner mitochondrial membrane. But of course, that membrane does not exist in a prokaryotic cell. So where is this machinery located? It has to be a membrane. Like I told you before, electron transfers are typically done by machines that are situated in membranes to keep them less like moving around, basically to stabilize them. So what membrane are we using in a prokaryotic cell? Right, we're using the plasma membrane, the cell membrane. In a eukaryotic cell, the protons are pumped across that inner mitochondrial membrane into the intermembrane space, and they're held in that nice little pouch, nicely isolated from both the cytoplasm and the matrix, which is good news because that now becomes a low pH environment, and that acidic environment can denature all the enzymatic processes that are happening in the cytoplasm and in the matrix. But we don't have access to that space in a prokaryote. So where do we put our hydrogen gradient? It has to form on either one side or the other of the cell membrane. So we've only got two options. We've got cytoplasm and we've got extracellular space. So where are we putting our proton gradient? Right. Yeah, it goes outside. We pump them outside. And this is good news because if we pump them inside, what we would have is quite a situation, right? The cytoplasm would become acidic, glycolysis would stop working because all the enzymes would fall apart. Krebs cycle would stop working for the same reason. Okay, last comparison. We said that in eukaryotes, we make 36 ATPs on average per glucose. How much ATP is made in a prokaryote? Is it the same? Is it less? Is it more? Are prokaryotes worse or better than you? Great. 38 prokaryotes are better than you. We've established that. Okay. So let me ask you this. Raise your hand if you have an answer to this question. Oh, before I do that, Paisley, it looks like you have a question. What was your question?
Is the acidic region the acidic compartment? Like on my paper, it says acidic compartment. Is that the same thing as acidic?
Yes. Yeah, and it's more accurate to call it a compartment when it's actually bounded by membranes as it is in eukaryotes, and it's a little bit less accurate to call it a compartment when it's prokaryotes. So I apologize for that semantics, but that's why I call it an acidic region sometimes. But that's a great question. Okay, so now raise your hand if you have a question, an answer to this. Why is it that prokaryotes make more ATP than you do? Amber. Uh-oh, you're muted.
Sorry, because it costs 2 ATP to transport the pyruvate through the mitochondrial membranes.
Correct. Exactly. And since we don't have to do that in a prokaryotic cell, we get a little bit more ATP. All right. So I'm going to show you a little cartoon that I drew. I think it will become very evident that I drew it. So we're going to see kind of how this all works in these different cells. So here we're seeing a prokaryote go through this glycolysis process, the Krebs cycle, electrons being removed by NADH, and then these NADHs are being oxidized back to NAD+ by dropping their electrons off at the electron transport chain, pumping protons into the extracellular space. Those protons come back through ATP synthase, ATP is made. In a eukaryote, we have a much more complicated situation. So here is the cell membrane. Here's a ginormous, unrealistically sized mitochondrion with 2 membranes, outer and inner membrane, intermembrane spaces here. Glycolysis is happening in the cytoplasm. Pyruvate is made. We get ATP production. We get NADH production. We get pyruvate moved into the matrix, costs us 2 ATPs, basically the glycolysis ATPs. We get decarboxylation to acetate. We got the Krebs cycle, lots of NADH is produced, CO2 is produced also in all of these. We get NADH being re-oxidized back to NAD+ by dropping its electrons at the electron transport chain. The electrons then are dropped onto oxygen, producing water. Hydrogen ions are pumped into the intermembrane space. They come back through ATP synthase and we get ATP production. So all of this is happening over here. CO2 is also being released by the Krebs cycle. And we also are having water production when oxygen is reduced. But we do see also the comparisons, the differences. All right. So that is prokaryotic versus eukaryotic aerobic cellular respiration. Oh, thanks, Alex. It's kind of a hot mess, but I appreciate you. All right, so again, that was aerobic cellular respiration in comparison between the prokaryotes and the eukaryotes. Now we need to make another comparison, which is between aerobic and anaerobic cellular respiration. And again, I want to really emphasize that anaerobic respiration, being anaerobic, does not require oxygen, but it is only one form of anaerobic metabolism, and fermentation is another form, and these are separate. And quite often by accident or by ignorance, they're conflated. So we're going to compare aerobic and anaerobic respiration. These pathways are similar in that they all do first glycolysis, the Krebs cycle, and then the electron transport chain. However, how are they different? We talked about this last time. Let's just see in the chat how they are different. Great. Yes, so it's in the terminal electron acceptor, the last man on the totem pole at the end of the electron transport chain. The last guy hands off the electrons to someone. That is the terminal electron acceptor. And that's oxygen in aerobic respiration, and it's not oxygen in anaerobic respiration, whereas some people have said, so they abbreviate terminal electron acceptor as TEA, like the drink. The T is different. I like that. Okay. So instead of oxygen in anaerobic respiration, you often have something like nitrate, sulfate, carbonate, or similar. Another molecule that's very electronegative, an inorganic electronegative molecule. So the important thing to know about that is that oxygen is more electronegative, so it's always a better acceptor. But you know, for some organisms, they're just not equipped to do aerobic respiration. And when you do anaerobic respiration, you get a lower ATP yield. And I can't give you a precise number because the different forms, and there's many, nitrate, carbonate, sulfate, other eights, whoopsies, they have different amounts of ATP. So all you need to know is that it's lower than the total number you can get. And typically anaerobic respiration is something performed by various kinds of prokaryotes. It's not found very much in the eukaryotic world. So we're just going to say it's a prokaryotic thing. And so the number you're comparing it to is less than 38. So now I'm going to have a little cartoon for that. Okay, so here is respiration. Okay, when we say respiration, whether it's aerobic or anaerobic, then we're talking about glycolysis, where glucose is broken down to 2 pyruvates, and that is then converted to acetate. And in that process, ATP and NADH is made, and in this process, a little bit of carbon dioxide is made. Then this acetate goes into the Krebs cycle, more ATP is made, more NADH is made, and more CO2 is released. NADH is oxidized back to NAD+. Those electrons flow through the chain. Their energy is used to pump protons to the outside. Notice that this is a prokaryotic cell, right? And then they come back through ATP synthase to make ATP. What isn't on here is the details of the terminal electron acceptor, because that differs between these two forms of respiration. So here it is for aerobic, obviously. The electrons are added to oxygen. That causes oxygen to become temporarily negative, and nearby protons that are floating around, because not all the protons are out here, although there's lots more, but there's going to be some in here too, they get attracted to the oxygen, and that forms water. Now, if we're talking about anaerobic respiration, then this final guy here is not actually there. There's somebody else. And here's just an example of nitrate reductase, who takes the electrons and puts them onto nitrate instead and forms nitrate. And so this different enzyme is responsible for doing this job. You could have a different enzyme that does carbonate reduction or sulfate reduction, et cetera. And again, just to hopefully hammer this point home, oxygen is a better acceptor. And so it is, the electron transport chain is a little bit longer for the electron, for the aerobic respiration version. And the electrons that arrive at oxygen are a little bit more de-energized. Nitrate isn't as good, carbonate isn't as good, sulfate isn't as good. So this guy is taking the electrons earlier and handing them off to the terminal electron acceptor, which means there are fewer opportunities to pump protons. You'll see that there's fewer opportunities. Here there's 3, here there's only 2. And so consequently, you don't get as much of a proton gradient formed and you don't get as much ATP. All right. So aerobic cellular respiration yields 38. Anaerobic, does it yield the same, more or less? This is basically a were you breathing and alive for the last 10 minutes of this class. And if you had, if you missed something because you were off, whatever, your kid was banging at the door, you had to take a pee break, you're having a bad morning, no judgment at all, but we've all been there. Okay, yeah. Anaerobic respiration yields less than 38 in a prokaryotic cell. And like I said, we're not going to really deal with a eukaryotic setting because that's not very relevant. Raise your hand if you'd like to explain why. Why is it that anaerobic respiration yields fewer ATPs? Amber, go ahead.
I think it's because oxygen is better, so it can produce more.
Yes. Does anyone have anything else they want to add to that? That's absolutely on the right track. Dylan, go ahead.
Yeah, I think you just said that essentially the electron transport chain is shorter, so there's less of a proton gradient on the outside, and since it takes the electron early, the electron that leaves in anaerobic respiration is higher in energy, so it makes, is it the AT synthase pump less efficient, or is that just okay?
It doesn't really have anything to do with this per se, but it's exactly what you said, and also what Amber said, which is oxygen's a better electron acceptor. So if you want to use these other guys, you have to take the electron earlier when it still has a little bit more energy in it. And that shortens the chain. It causes fewer opportunities for proton pumping because the chain has multiple opportunities for proton pumping. That causes the gradient to be smaller. And as a consequence, then the number of protons flowing through here just isn't as many. So you don't get as many turns of this little machine here to produce ATP. And while this is detail that's not necessary, I'll just say this for those of you who sort of need more detail to understand something. This is like a turbine. It's not very dissimilar from the turbine of a dam when water flows through it to generate energy in a dam. The number of protons flowing through here causes this to spin a certain number of times. So if you have fewer protons flowing through, this spins less. And the spinning kinetic energy is what generates ATP. Okay, another comparison, respiration and fermentation. So again, we really have to nail down this difference that aerobic and anaerobic respiration is very distinctly different from fermentation. Now, what do they have in common? Both respiration, whether it's aerobic or anaerobic, and fermentation, both start with glycolysis. The glucose molecule is broken in half, some ATP is generated, some electrons are taken out and made into NADH. But there are some important differences. Put that into the chat, or if it's too long of an answer for you to put in chat, you're welcome to raise your hand as well. Okay, Athena says that fermentation produces less ATP than respiration, and that is true. Great. And then Karen says there's no Krebs or electron transport chain, and therefore there's a lower ATP yield. And Dylan said something similar. Yeah. That's right, Annika. Fermentation, basically, glycolysis is the first part of it, and then the so-called fermentation reactions are really just restoring NAD to feed back into glycolysis so they don't produce any energy on their own. Great. Okay. All right. Yes, Amber, good point. The terminal electron acceptor is different. In respiration, the terminal electron acceptor is always an electronegative inorganic molecule like oxygen or oxygen equivalent. And in fermentation, the terminal electron acceptor, the place you put the electrons that you took out of glucose, is an organic molecule. And in fact, those electrons are not used to make energy. They were taken out and then you put them back onto something else. So it's like you didn't get any benefit from them. Okay, so the major difference is that fermentation basically involves glycolysis and then a little side reaction just to restore the tools that you need for glycolysis. Whereas respiration goes through glycolysis and then the Krebs cycle and then the electron transport chain. And so energy from electrons is used to power ATP synthase. That doesn't happen in fermentation at all. And as Amber mentioned, the terminal electron acceptor is different. In respiration, it's an inorganic molecule that's electronegative like oxygen or something similar. And in fermentation, it's organic, an organic molecule, something that's got a lot of energy. Fermentation, as many of you mentioned, has a much lower ATP yield, and it's always the same. There's billions of types of fermentation, but that doesn't make any difference in terms of the energy yield because all the energy is being made in glycolysis, which is 2, as all of you have memorized probably if you were woken in the middle of the night and someone said, how many ATPs is produced in glycolysis? You'd be able to answer that question. Fermentation has 2 ATP production, and then the other reactions that are considered part of fermentation are not energy producing. So let's have a little look at that. So here is basically respiration, right? Whether it's in a eukaryote or a prokaryote, whether it is aerobic or anaerobic, glycolysis is happening, and in that process, we get some electrons taken out and made into NADH. We have decarboxylation of the pyruvate, which also involves removing electrons and throwing away CO2, that's not shown here. We have our Krebs cycle where more NADH is produced. We go to our electron transport chain and all these NADHs come barreling through and drop off their electrons and oxidize NADH back to NAD+, so that NAD+ can go back to the Krebs cycle, back to this intermediate step, back to glycolysis. And through the process, a very complicated process of the electron transport chain, you get a lot of ATP produced. So that's respiration. You know, the details here make it aerobic or anaerobic, but this is basically what's happening. Let's compare that to fermentation. So fermentation, we go through this part, glycolysis, and then we just take a detour. Right, so we still make NADH because we still take electrons out. In the process of breaking glucose, it's sort of required to take some electrons out. That's just how the chemistry works. But now you've got this NADH, and it actually can't go to the electron transport chain, and there's no Krebs cycle taking the pyruvate because all of the enzymes down here are missing. And so instead what happens is this NADH is oxidized back to NAD+ so that it can go to a new glycolysis cycle. And you have to put those electrons somewhere. Remember, redox reactions are paired. So pyruvate is the acceptor. Pyruvate is the terminal electron acceptor, and it turns into lactic acid. This is called lactic acid fermentation. That's what happens in our bodies. You know, a little bit. Mostly this is what happens, but there's a little bit of this. Now, some forms of fermentation, because this is just one of like a billion forms of fermentation, they go through this other step 2, where pyruvate is decarboxylated and CO2 is released. This is the gas part of fermentation. And then instead of putting electrons onto pyruvate, you put them onto acetate. And now you still get back your NAD+. The reduced form of acetate is ethanol. That's ethanol fermentation. And this is done in yeast, which makes beer, wine, and bread. And it's also why your beer and wine is fizzy at first. Now, there are many, many other variations on this theme where there's some kind of, whether you go this way or down here and then across, depends on what enzymes you have as an organism. So we have the enzyme to do this step. We don't have the enzymes to do this, otherwise we'd all be drunk on our bread, basically. And other organisms might have this enzyme and that enzyme, but not the rest of the stuff down here. And there may be other enzymes, like there could be something that takes pyruvate and makes something else instead of lactic acid, or something that takes acetate and makes something else instead of ethanol. All right. So this is another picture that I did not draw, so it's much nicer. And you can see glycolysis, and then heading off in this direction, the Krebs cycle electron transport chain for respiration. Glycolysis heading off to a non-energy producing reaction for fermentation. Okay. The other point that I make in the video lectures that you'll already have watched is that, as I said before, there's lots of forms of fermentation. Depending on which enzyme an organism has, it can use that pyruvate that it makes after glycolysis to make all kinds of things. Just depends on what enzyme it has. So this is how we do it. And this is also how Streptococcus lactobacilli, the one that makes yogurt, and various bacillus species do it. Ethanol fermentation is performed by Saccharomyces cerevisiae, the yeast that makes beer, wine, and bread. But you can see there's a whole bunch of other options. For instance, propionibacteria make propionic acid, which makes Swiss cheese have that unique taste that it has. And Clostridium, that anaerobic genus that has all kinds of pathogens in it, like Clostridium tetani, Clostridium botulinum, etc., makes a bunch of organic solvents. That sounds yummy. And then we've got a variety of other options. Just checking our time. Okay. So all of that has to do with the catabolism of carbohydrates. We want to take a minute to appreciate that, of course, proteins and lipids can also be used for energy, as we all know. And so let's look at where that happens and how it works. So here is another line diagram of respiration, basically. And you can see that it starts with glucose. Here is a triglyceride, right? 3 fatty acids attached to a glycerol. This is the fat that we usually, when we consume food and there's fat in it, it's in this form of triglycerides. That's the storage form of fat that's found in plants and animals. So the first thing we have to do is break it into pieces. So we take the glycerol off. The glycerol is 3 carbons. So you put that into this whole process at about the level of the 3-carbon pyruvate. Then you remove each of the fatty acids, and the fatty acid tails, if you remember this from chemistry, maybe, are always paired in an even number. So typically they're like 16 or 18 carbons long. And so we chop them into their pairs, and these 2-carbon pieces are basically acetate, and you throw them into this step of respiration, and they just crank out lots of NADHs in the Krebs cycle. So that's how fats are metabolized. Proteins are long chains of amino acids. The amino acids look like this. There's a central carbon, there is a carboxyl group, an amino group. The amino group has nitrogen in it. That's unhelpful, so we chop it off and throw it away. That's called deamination. And then this either has 2 carbons or 3 carbons, depending on what the R group is, or 4 or 5 or 6. So if it's just 2 carbons, we can actually put it in here. If it's 3 carbons, we can put it in here, and it's 4, 5, or 6, we can put it in the Krebs cycle because there are 4, 5, and 6 carbon intermediates in the Krebs cycle. So this is a really quick and dirty summary of how protein catabolism works. But first you have to chop the amino acids apart. That's proteolysis. Then you have to deaminate them. That's removing the amino group. You can see it's all gone. And then you have to put these different amino acids into different places depending on how many carbons they have. Okay. I asked you to think about this at the very beginning. So this is all about exoenzymes. You and other multicelled organisms have a digestive tract usually, especially animal-like organisms. And that digestive tract, as you eat food, it breaks the food into pieces, right? It breaks the proteins into amino acids. It breaks the fats, the lipids into fatty acids and glycerol. It breaks the carbohydrate chains, which most of the carbohydrates in the world are like complex carbohydrates and chains. It breaks them up into monosaccharides. But bacteria can't do that. So what do they do instead? They take the digestive enzymes that are in your intestines and they spit them out into the world. They digest the world and wait for the pieces to come back to them. So if you want to break starch into glucose, what enzyme do we use to do that? We eat starch, we eat vegetables and vegetable-based things. It has starch in it. We break it into glucose with an enzyme. What's that enzyme called? This might have come up in A&P. If you've taken nutrition, it will have come up. Yeah. The enzyme that breaks starch into glucose is called amylase. And bacteria have this too. They spit amylase into the environment, right? So they spit into the world, like literally, and then digest the carbohydrates. And then the pieces of glucose come back and they import them with glucose transporters. They also secrete enzymes to digest triglycerides, that, those 3 long chain fatty acids attached to glycerol, into fatty acids and glycerol. It's basically a lipid. You need an enzyme to digest a lipid. So if you were going to name an enzyme that digests lipids, what would you call it? Yes. When we do it, which happens in our stomach and our small intestine, it's called lipase. And when bacteria do it, it's also called lipase. Okay. Protein needs to be broken into amino acids. We have proteases in our stomach and in our small intestine to do this, and so do microbes. Okay. I know it's a lot of chemistry. Take a breath. Is this part of why certain bacteria harm us? Because they're, yes, Annika, actually, they are sometimes harmful because they are secreting enzymes that can do damage to our bits and pieces, although it's more the case that that's happening if they get inside the body where they're not supposed to be, because when they're coming through the GI tract, we have this thick mucus layer that's protecting us from bacteria. And then in the stomach, there's this low pH that kills most bacteria because most of them can't survive, although a few can. And that pH also caused their own proteins and enzymes to fall apart. So their enzymes are degraded by our pH in the stomach. The acidophiles like Helicobacter pylori that can survive in low pH are particularly nasty. They also have a really fun lot of flagella, and they burrow into your mucus and cause ulcers. But most bacteria don't do that. However, when they get into if they get subcutaneous or if they get into organs and stuff where you don't have those kinds of protections, yeah, they can do a lot of damage. All right, so let's talk about photosynthesis. We've talked about catabolism a lot. We're going to talk about the autotrophic reactions, that carbon fixation reactions. And actually, before I do this, let me kind of check in with you guys. We're about 6 minutes away from where I would normally take a break. Are you okay to do photosynthesis before the break, or do you need your break now? So wait and work, or break now. Just put that into the chat. Okay, wait and work. All right, let's do photosynthesis real quick, guys. We can do this. All right, now let's focus on eukaryotes. And remember, this is part of the worksheet I had you do. Where in eukaryotes do the light reactions happen? Specifically, where are your photosystems that are collecting sunlight and the associated electron transport chains that are leveraging the energy from those energized electrons of the photosystems to get energy? Where is that located? The light reactions are happening where? In a eukaryote, so you can assume it's happening in the chloroplast. Yes, the thylakoid membrane, exactly. Right, so remember, a chloroplast has an outer and an inner membrane where nothing particularly interesting happens, and it has a third internal membrane called the thylakoid membrane surrounding these little pouches, and it's studded with photosystems and electron transport chains. Now, the electron transport chains are doing something very similar to what happens in respiration, which is that electrons are fed to them from the photosystems, and the energy from that electrons is used to pump protons, and the gradient accumulates where? Exactly. the inside of the thylakoid, called the interior of the thylakoid or the lumen of the thylakoid. Okay, in the light reactions, those high energy electrons energized by the sun, are fed through the electron transport chain. Hydrogen ion is formed, hydrogen ion gradient is formed. It is allowed to come back through ATP synthase, so ATP is formed by the light reactions. Then the dark reactions happen. It's called the Calvin cycle. That's where the energy that we just made is used to make sugar. Where's the Calvin cycle taking place? Again, it's a cycle, kind of like the Krebs cycle, with a bunch of enzymes that goes in a circle. Yes. The interior fluid bathing the thylakoids, not the inside of the thylakoids, but in the chloroplast, the inner and outer membranes contain a fluid called the stroma. And that's the fluid that's bathing the thylakoids. Okay, so this summarizes what you've just told me, okay? That the light reactions happen in the thylakoid membrane, the Calvin cycle happens in the stroma, which is outside of the thylakoid, and the acidic compartment is inside the thylakoid, which is good news because the stroma and the acidic compartment are separate from each other. And so the enzymes here, really important for the Calvin cycle, don't get denatured. Let's talk about prokaryotes. They don't have chloroplasts, but some of them can still do photosynthesis. Where do their light reactions happen? There's no thylakoid membrane, so what membrane do they use? Right. Yes. They use their plasma membrane. So they stick it in the plasma membrane, just like the bacteria that can do respiration stuck their electron transport chain machinery and their ATP synthase there. Where's the Calvin cycle happening? There's no chloroplast, there's no chloroplast stroma. It is a fluid. Where's the neutral fluid that you can put your Calvin cycle into? Right, exactly. It's happening in the cytoplasm. Okay, one thing that's really important that happens in the light reactions is that we form a proton gradient to power ATP synthesis. So where are we putting our protons? We can't put them in the interior of the thylakoid, which is a nice safe little pouch that is separate from the stroma and the cytoplasm. So where do we put our hydrogen ions during photosynthesis? Yes, the same place we put them during respiration, right? Outside the cell. Exactly. Good. So now I'll do a little cartoon. Let's start with eukaryotic photosynthesis, right? And we've got this unreasonably large chloroplast, just so it's easy to see everything. It's got two membranes, outer and inner, where nothing happens. And the thylakoid, there's one thylakoid. There's our photosystem taking electrons off of water. Those electrons are energized by the sun. The electrons flow through the electron transport chain. Protons are pumped to the interior of the thylakoid. The electron goes to the second photosystem. It's re-energized by the sun. It's then collected by NADP and makes NADPH. We get ATP production. The Calvin cycle is happening in the stroma. These are the dark reactions. And what it's using is all of the energy, ATP, electrons, and CO2 to make a 3-carbon sugar. And then we put 2 of those together in a separate reaction to make glucose. All right. Just a reminder, if you didn't cover this very thoroughly in cell biology, NADP is very similar to NAD+. It just happens to be the molecule that's used in photosynthesis as an electron carrier. And NADPH is similar to NADH in that it is the taxi cab full of electrons. Let's look at what happens in a prokaryotic cell. So we have just one membrane, cell membrane. That's where the photosystems are. They're being energized by light. The electrons are being stolen from water, making oxygen. This is why oxygen is produced. And then they're energized, put through the electron transport chain that's sitting in the membrane. Protons are pumped outside, and then the electrons from this chain go to the second one, re-energized, and then they're placed onto NADP. So we have NADPH carrying these high energy electrons. All these external protons come back through ATP synthase to make the ATP we need, and the electrons we need, and the CO2 we need to do the Calvin cycle reaction to produce this 3-carbon molecule, and 2 of which produce glucose. Okay. One last little thingy, and maybe we'll do this cycle comparison after the break actually to kind of see if we can re-energize those thought processes. So we'll take our break now, and I'll see you in 9 minutes at the top of the hour at 11 o'clock. Thank you for waiting, and we'll do a little bit of wrap up. Question, Anthony, go ahead.
This isn't like a question kind of not relating to this stuff, but so I'm gonna be out of.
All right. I was just reading the chat here, and I love Annika's analogy here. I feel like the eukaryotic prokaryotic difference is like the difference between a purse with a lot of pockets and a big hobo bag. I love that. That comparison is great. Okay, let's talk about it. So now what I'm doing is I'm comparing completely different processes, or at least things that are apparently very different that you probably never put in the same category as each other. When we think about respiration, one of the key features is that Krebs cycle, where all the electrons are taken out of the organic molecule until all that's left is carbon dioxide that gets thrown away. And in the Calvin cycle, the opposite is happening. You're putting carbon dioxides together to ultimately to make glucose. So if we compare these cycles to each other, we can compare them in lots of ways. Let's start with where they each happen in eukaryotes and prokaryotes. So in eukaryotic cells, both the Calvin cycle and the Krebs cycle happen inside of a specialized fluid inside of an organelle. In the case of the Calvin cycle, it's happening in the stroma. And in the case of the Krebs cycle, it's happening in the matrix. But both of them are slightly basic, neutral to basic, pH of around 8 fluids inside of a organelle. Whereas in prokaryotes, both these cycles are happening in the cytoplasm. All right, let's do a little chat voting here. So let's start with the Calvin cycle. Do you think that the Calvin cycle, where carbon dioxides are funneled in and then electrons are added to them in order to construct a 3-carbon organic molecule, is that pathway overall oxidizing or reducing from the perspective of the cell? You know, sort of, yes, bearing in mind that everything is both, but in terms of what we get out of it as a cell, which is the 3-carbon organic molecule, is this pathway overall oxidizing or reducing? Yes, good job, you guys. You were right on that. You were like, putting electrons onto carbon dioxide, reducing. Correct, that's exactly right. Let's look at the Krebs cycle. In the Krebs cycle, what happens is that a 2-carbon molecule comes in, and then we mess with it a little bit and kind of tear off both of its legs and throw them away as carbon dioxide. But in the meantime, we take out a lot of the electrons. So is this pathway or this cycle overall oxidizing or reducing? Exactly. In terms of what's happening to the organic molecule, it's being oxidized. Of course, it would be true to say that NAD is being reduced to NADH, but we don't care so much about NADH. We care about what's happening to that organic molecule. Okay, good job. So in the Calvin cycle, carbon dioxide is reduced, electrons are added to make an organic molecule. And in the Krebs cycle, the organic molecule that you start with is oxidized by having its electrons removed, and then the carbon dioxide that's left is thrown away. So for each pathway, there's a big picture purpose, right? The Calvin cycle's purpose is to do something called carbon fixation, where you take CO2 and you make it into an organic molecule. You're fixing carbon. That's just a funny term that's used in the field to talk about when you take an inorganic molecule out of like the air. In some cases, it's out of soil when it's nitrate fixation, but in any case, and you make it into this larger complex organic molecule that organism and other organisms around it can use. Carbon fixers are really, really important for the ecosystem. And the Krebs cycle, the purpose of this pathway is to oxidize, or we could slangily say burn, organic molecules for energy. So they're kind of the opposite of each other, right? Okay, now if we're going to focus on carbon fixation, photosynthesis is one of the major carbon fixation pathways in microbes. What's the other one? Put that into the chat. And yes, please use your question mark if you don't remember. That's absolutely fine. This is going to be the harder one to remember because you almost certainly will not have heard of it before this class. And it's a long, kind of convoluted, unwieldy term. There we go. Yes. So far, my favorite answer is chemo something something. Chemolithoautotrophy. Let's break it down. Chemical. Litho means rock. Autotrophy means is just a term that literally is the wrong word. It's self-feeding, but it means that you're able to make your food, your glucose, out of raw materials like CO2. Yes, chemical rock eaters. Good job, Dylan. So what happens in chemolithoautotrophy? So let's have a cartoon about that. We start with something like, this is just one possible version of this pathway, something like an iron atom, an iron ion, I should say, that has some disposable electrons. We remove one of the electrons. So this is an example of an iron 2 positive ion, and we take an electron off, and now it exists in this other ionic form. So you really need something that can do this. Not a lot of things can do this. That electron is fed through an electron transport chain. The energy is used to pump protons, creating a gradient. That gradient is then leveraged to create ATP through ATP synthase. And we get ATP plus some extra electrons plus CO2, and we put together a 3-carbon molecule, which is then 2 of these are smushed together into glucose. Why are chemolithoautotrophs important? Raise your hand. I mean, this is a cool, funky, wackadoodle pathway. Why should we care about it? Or why does anybody, any living thing in the world care about it? Good. Okay. So good answers are showing up in the chat. They do carbon fixation. They help produce glucose, basically glucose. Carbon fixation is the production of glucose from CO2 in areas that don't have access to sunlight. So photosynthesis is doing the same job in the sunlit areas of the world where plants and algae and photosynthetic bacteria produce glucose so that the rest of the organisms on Earth can eat it. Not just that, they basically make more than they themselves need, though they also need it. But there are also forms of life that exist below the surface, so in soil, deep in the soil where the sunlight doesn't penetrate, deep in the ocean where the sunlight doesn't penetrate, or underground caves and mines and places where there's no sunlight. There's still creatures living. This is why we call them the photosynthesizers of the dark. They're not doing photosynthesis because that implies light, but they are doing chemolithoautotrophy, which gives the same result without light. And here's just a picture of one of the environments where you will find these guys deep in the ocean. We already took our break, sorry. I asked you this question last time. Let's see if we feel a little more confident this time. Aerobic cellular respiration is overall oxidizing or reducing? Excellent. Yes, glucose is oxidized by having its electrons removed. The product is CO2. Those electrons are used to make energy in the form of ATP ultimately. Anaerobic cellular respiration, overall oxidizing or reducing? Excellent. I want to remind you, I don't know if you'll remember this, that last time your first instinctive answer was to say it was reducing, it must be different. And now you're saying it's oxidizing, which is accurate, okay? Because the same thing is happening. Glucose is being oxidized to CO2. It's, there's some like detailed differences in terms of who's the terminal electron acceptor, but who cares about that? I do, but you don't need to in this question. Oxidizing is the answer. Now, fermentation was that funky one, right? Where the first part, glucose oxidation to pyruvate, is oxidizing, but then pyruvate's reduced to acid. So it's kind of both and neither at the same time. We sometimes still refer to fermentation as an oxidation reaction because of this first part. But really, it's just not very accurate because we're not, the energy that we get is at substrate level phosphorylation and glycolysis. And it's really, we're not using the electrons for energy. Photosynthesis. Yes. Good job, you guys. Carbon dioxide has electrons added to it, plus some other bits, and that forms glucose. Chemolithoautotrophy. Yeah, because it's basically the same, right? Some detailed differences in terms of where the electrons come from. In this photosynthetic world, the electrons come from water and they're energized by sun. Here, they typically come from some inorganic substance like an iron atom or sometimes, sulfur, other kinds of things. Nonetheless, it's reducing because you add those electrons to CO2 to make glucose. Okay, I asked you about a flow chart last time. What is this electron flow chart depicting? Which process above? You go through glycolysis. The electrons are removed in glycolysis, they're removed in the Krebs cycle, they end up in the electron transport chain and they're put onto nitrate. A lot fewer question marks today, notice that? A lot of you, not everybody, so if that's not you, you can review this stuff and get more confident, but a lot of you are feeling more confident that you understand, like, what am I getting at here? Anaerobic cellular respiration. And this one, an iron atom has its electrons fed through electron transport chain, put onto CO2, making sugar. Yes. And feel free to use your question mark if you're still not sure. That is totally fine. All right. Yeah, that one's chemolithoautotrophy. All right. I want you to take a second to make a carbon flow chart for fermentation and for photosynthesis. We're going to start with fermentation. So you're going to put that into the chat when you feel ready. I'm going to give you just a couple minutes to think about this. There isn't just one right answer. What I want you to think about is go through step by step through this process, not every single step, but like the major steps, and ask yourself where the carbon is at each step. So that's all the guidance I'm going to give you right now. Take a second for fermentation to write out the carbon flow chart. And I'll bug you in a second to put your answers in the chat. Great. Okay, so again, for fermentation, the carbon flow is from glucose to pyruvate. And then for lactic acid fermentation, at least, we reduce that pyruvate to an organic acid. And that's where the carbon is. It stays here the whole time. In photosynthesis, we start with CO2. That's the first step where we have carbon and we reduce it to glucose. So if you were tempted to write out the whole photosynthetic process, if I just ask you what's happening with carbon, you have to kind of leave out all the light reactions. Now, I don't want you to fret about this too much, okay? But we're doing this to kind of get a different perspective on these pathways. You'll notice that I'm not asking you to memorize all the detailed steps in each pathway. I'm taking kind of a much bigger step back to say, what's the purpose of these pathways? If we understand that, we can compare completely different processes to each other and see the interesting things, like one is actually sort of the backwards version of the other one. We also want to apply our metabolic learning to the lab setting, to the unknown ID process, and also to the growth and testing that is done by real-world microbiologists to understand microbes and also to classify and identify them. So here again is our thioglycolate test that tells us whether an organism is capable of aerobic respiration, whether it's capable of anaerobic metabolism, and what kind of enzymes it might have. So we can learn a lot from this test, actually. If we look at these tubes of thioglycolate results, we can see that it's telling us that this one and this one are capable of doing aerobic respiration because they're both thriving at the top where there's lots of oxygen. This one can't, and this one doesn't. It's alive in oxygen, but it's not doing aerobic respiration. Let me ask you this. How do I know that? How do I know this is a minus and this is a plus? Raise your hand. Annika, go ahead.
It's not clumped up by the highest oxygen area. It's indifferent to it.
Yes. If this was doing aerobic respiration, and we can see that it's also growing anaerobically, then whatever process it's doing down here, it's either anaerobic respiration or fermentation. And we already know that those processes don't produce as much ATP, so growth will be slower. And if it was doing both things, you would see something more like this, where there's some growth down here because anaerobic respiration or fermentation is taking care of business. And there's lots of growth up here because aerobic respiration is producing more ATP, therefore faster growth. But that's not what we see here. We see just an even amount of growth. So whatever metabolism it's doing, it's the same all the way through the tube. Which of these can do anaerobic metabolism? Which of these tubes? Write in the chat using these letters, which of these tubes show that the microbe can do anaerobic metabolism? Good. Okay, so a lot of you picked this one. That's obvious, right? It's the only thing it's doing, and it's not even alive without presence of oxygen. But we can see that both these tubes are capable of anaerobic metabolism as well. They can grow in oxygen. They even could use oxygen in this case, but they are able to grow without it. Which of these tubes is doing fermentation? Good. Good. I love it. You guys, you're so on this. You have gotten used to using your question mark strategically. Good job, because sometimes the answer is a question mark. We don't know. Okay, why don't we know? Raise your hand if you can explain to me, why don't we know the answer to this question? Amber, go ahead.
I think it's because we don't know what kind of respiration or fermentation is being used. We just know that it doesn't use oxygen.
Yes, because
I think that came out right.
Yes, no, absolutely right. Because we know it's growing down here, so it's doing some kind of anaerobic metabolism, but I have bashed you over the head to remind you that there's several forms and there's nothing to tell us which form. So is it fermentation? Maybe, but it could be anaerobic respiration, and we can't tell that from these pictures. So these are all question marks. Maybe is the answer for all of these. Definitely no for this one. All right, which of these tubes has the enzyme superoxide dismutase? And I will say that there is a way to tell, but you're welcome to use your question mark as in because you just don't know. That's totally fine. But in this case, there actually is an answer. And so you're welcome to take a stab at it, and you can question mark your stab at it too. Good, good, good. Okay. So a lot more uncertainty here, but I'm just going to remind you that this is an enzyme that's used by all organisms that are able to survive in oxygen. If you're living in oxygen, you have this. End of story. Okay, so that's these three guys. Yes. What about catalase? Not as straightforward. Okay. So this is another case where the question marks are king. I don't know if you're going to remember this, okay? But there's a couple of places where we can say definitely no catalase, okay? So the no catalase, for sure no catalase is this one, of course, but also arrow tolerance do not have catalase, okay? But these ones we can't be sure. And the reason we can't be sure is that even though it's possible, even maybe likely, there's another option. Some organisms have peroxidase that does the same job more or less. It does the same job in the sense that it detoxifies hydrogen peroxide. The specific details are different. So we don't know for sure if they're doing it with, we know they have something. It could be catalase, it could be peroxidase. It's probably catalase, but we don't know that for sure. We know this guy doesn't have catalase, and we know this guy doesn't have catalase because arrow tolerance, remember, they have SOD, they have superoxide dismutase only. All right, who's got a cytochrome C oxidase? Let's start with who we know doesn't have it. How about that? Who does not have cytochrome C oxidase? Yeah, confidence there, good. And to be honest, I think I have a typo in my, my diagram. We're about to find out if I fixed it yet or not. So yay, the suspense. So the guys that don't have it are going to be the ones that do not do aerobic respiration. So all of you picked up on the fact that that's D, of course, but there is another one that does not do aerobic respiration. Who is that? There we go. Right. So we know that A and D do not have it because they don't do aerobic respiration. And this is the final component of the electron transport chain in aerobic respiration for some organisms. Oh, I did fix my typo, yay. Okay, so cytochrome C oxidase is the primary component of the aerobic respiration transport chain, but there is an alternative, and so some organisms use that alternative instead. So we can be relatively sure that this has cytochrome C oxidase, but not 100%. And we can be relatively sure this doesn't, but not 100%. So it might, and this probably does, but might not. Okay. Who's got nitrate reductase? There we go. Don't be too confident. I mean, be confident, but not too confident. Okay. So a lot of you are guessing maybe A and D, but your question marks are justified. We don't really know, okay? We know that, again, just like we didn't know for fermentation, we don't know exactly what happens anaerobically here without more information, at least. As Amber said, we need more testing. We could do a nitrate test, that would tell us, right? All right. Good, we still have time for all the things. So what you'll notice, I hope, do you remember that thioglycolate actually officially is part of chapter 6, the growth section that was last week? But here we are bringing it back in to metabolism because it's relevant. And the kind of questions that you just answered, they could be on the test. So let's look at broth. So remember we had some broth tests for things that were growing in different conditions. This is a broth test that looks at oxygen metabolism, not using thioglycolate. Okay, so you're just taking broth and you put the microbe in it and you grow it either with or without oxygen. So clearly when there's no growth, whatever condition it's in, it's dead. And when there's a lot of growth, it was happy. And when there's medium growth, it was sort of medium happy. So we take two different mesophiles, okay, two different species that are mesophiles. We grow them in oxygen. One is a facultative anaerobe and the other is aerotolerant. You can assume the broths are fine, that we're not doing some sort of trivial thing where it's like they're fastidious, they're going to be dead in this type of broth. This is a broth they could use if the conditions are right. Now there's 3 options here, but there's only 2 species. So I'm giving you all the possibilities: grow really well, don't grow, or grow medium. Which of these tubes represents the facultative anaerobe grown in oxygen? And which one, the other one would be the aerotolerant. Maybe I'm going to ask this a different way because I think this question, just the way I've worded it, is a little bit confusing. So let me start over. Just clear your palate cleanser for the brain, put some orange sorbet in there or whatever. Let's ask which tube represents the aerotolerant one. Maybe that will be a little bit logically easier to answer. Which one represents the aerotolerant? One of these tubes is the facultative anaerobe, one is the aerotolerant, and there's one distractor. Okay. Yeah, you should be doubting your choices, guys. So let's work through it logically together because people are not there yet, but that's okay. Let's try to figure out which of these tubes we can eliminate. So based on the fact that the one's a facultative anaerobe, is this guy growing in oxygen? Is this guy growing in oxygen, this facultative anaerobe? Okay, yes, it can. What about the aerotolerant guy? Does that guy grow in oxygen? Yes. Okay, so is there one of these tubes that we can just disqualify off the bat and say it's not, whatever these guys are, it's definitely not this tube. Good. It's not C. These both can grow in oxygen, so we can eliminate this possibility. Bye-bye. Okay, so now we've got good growth and medium growth. So let's go back to my original question. Which of these tubes represents the facultative anaerobe? The other one would be aerotolerant. Which one represents the facultative anaerobe? Let's remember what facultative anaerobes do. They thrive in oxygen and they can get by without it. Aerotolerants, they grow in both conditions, but whatever they're doing anaerobic metabolism, so they don't do aerobic respiration. Good. Okay, so hopefully now you understand where I'm going, which is that this tube is obviously the facultative anaerobe because it's doing aerobic respiration. And this guy, the aerotolerant, is doing some kind of anaerobic metabolism despite the presence of oxygen, doesn't care about oxygen. And so this is growing faster. All right, let's do another one. Isn't this fun? I'm sure you were hoping for another one. I'm sure that was what you were thinking just now. A facultative anaerobe now, so the same species, just one species, but it's inoculated into two different tubes and grown for 8 hours at its favorite temperature. Just one of those tubes was grown in air and one was grown without air. Which of these tubes represents the tube that was grown with no air? Great. So once again, we know this facultative anaerobe can grow in oxygen, but it can also grow without. So we know that we can disqualify this tube. So now we have these two options. So we have to ask ourselves, since we clearly can see there's a growth difference, it's going to grow better in oxygen. So the one without air is going to be this one, A, which most of you got. Good job. All right, and let me just go back to this Y again. I've sort of said this many times, but it's kind of a difficult concept to master. So how do I know that this is the one in oxygen and this is the one in in anaerobic or no oxygen? How do I know that? And why is there this difference? Why is it growing slower with no oxygen? Raise your hand. Karen.
We know that aerobic respiration, since O2 is a stronger receptor or electron or whatever, that everything will grow faster in oxygen. It'll still grow without it, but it will be slower.
If an organism is doing aerobic respiration, it's producing more ATP per glucose, and so it's going to grow faster. Just as a real quick pulse check, just be honest. Did you understand that, yes or no? Good. Okay, so I'm just going to say this again, and then we can circle this back around to the Q&A. I'm happy to do that. Aerobic respiration produces more ATP per glucose, and an organism that's doing aerobic respiration is going to grow faster with all those extra ATPs than if it is doing anaerobic metabolism. So clearly, when you're comparing the same species grown in these two conditions, as long as it can grow both with oxygen and without, but if it's able to use oxygen, then it's going to grow faster in oxygen. Now, would this be different? Would these tubes look different if we were dealing with an aerotolerant organism that was grown in both oxygen and no oxygen? That's right, Alex. Yes. A facultative anaerobe can do both, but grows faster with oxygen because it's doing aerobic respiration. An aerotolerant, so now I'm just changing this scenario. If we were dealing with an aerotolerant organism and we put it in one tube in air and one in no air, would we still see these two different results? They would look the same. That's right, Amber. They would look the same, right? Because in both conditions, it's doing the same metabolism. Okay. Now, I would say, based on my pulse check, maybe 60% of you are feeling super confident. Some of you are sort of grasping it, not quite, and others are feeling still a bit lost. So I do want to make sure to get through the rest of my slides, but I'm happy to revisit this because we do have time in the Q&A. So just hang tight for that. I want to talk about the metabolic and growth tests that you're going to need for your unknown. I just want to give you this laundry list. I'm going to talk about this again in lab meeting. And so for those of you I'll see in just a half an hour, or those of you who I saw last night, or those I see on Saturday, you'll get this again, kind of. But there's a multiple growth and metabolic tests that you can and may end up doing for your unknown. So lab 4 is kind of, the purpose of lab 4 is to kind of get your feet wet in that arena, but you're going to have to do a little bit of research and understanding and analysis on your own with my help as much as you need for these other metabolic tests. And so what you have access to is broth tests at all kinds of temperatures, all kinds of pHs, all kinds of salt concentrations. You have access to the oxygen preference thioglycolate test. There's a bunch of enzyme tests that include catalase and oxidase, but also nitrate reductase and sulfate reduction and all kinds of other enzyme tests like amylase and caseinase and lipase. And you have a bunch of metabolic tests that ask whether a particular sugar can be fermented. These are the phenol red sugars. And then if your organism happens to be an obligate aerobe, you have access to some other sugar tests, but mostly you will not need those. In the lab meeting, I talk about catalase testing. I just want to remind you what a positive and negative catalase test looks like. But let's see if we can have a little sort of analysis here. You have a facultative anaerobe and an obligate anaerobe, which gave each result. So let's start with, so let me see if I can make this more clear. I feel like my wording was just super confusing. Sorry about that. Okay, there's two species, A and B. They're both tested with catalase. This is clearly a positive result. This is clearly a negative result. But one of these species, A, gives this result, and the other species, B, gives this result. And these two species are, one of them's a facultative anaerobe and one's an obligate anaerobe, but you don't know which is which. So which one is A? Is it the facultative anaerobe or is it the obligate anaerobe? Excellent. And so I'm poking at this from another angle that I could ask you on a test. And so now you don't have to worry about whether or not your facultative anaerobe has catalase, right? Which if I was asking you a different way, you wouldn't know. Now I'm saying one of these guys has catalase. Who does it have to be, right? If you only have two options, it's going to be the facultative anaerobe. Whoops. Because the obligate anaerobe definitely does not have catalase. All right, so now let's look at the oxidase test. This is a test many of you will do in the course of your unknown ID. Here's the positive on the left, negative result is on the right. Yours, when you receive it, will be probably on a circular paper, but the color is really the important part. If an unknown gives you a positive result, what can you conclude? Write that into the chat, what you conclude from the positive result. Anything that you feel confident you can say based on this result. And just to be clear, we're not talking about the same organisms as we were just talking about before, and I apologize if that was unclear. Okay, good. Good. Lots of different answers that are all accurate in the chat, and also lots of question marks that are also welcome. Okay, so let me read off some of the correct answers. The first and most obvious and straightforward conclusion is that this oxidase test specifically asks, do you have cytochrome C oxidase? The answer, if it's positive, is yes, you do. If an unknown gets a positive, then you can say with confidence, this has cytochrome C oxidase. The next thing you can sort of extrapolate from that is, that means it definitely can do aerobic cellular respiration, and it's able to survive in oxygen, in fact, thrives in oxygen. Okay. Now, if it's negative, what can you say? You can say it doesn't have that enzyme, but it doesn't tell you a lot of other things because there's other enzymes that can do the same job. All right, looking at a nitrate test. There is a two-step process for the nitrate test. If you haven't read lab 4 yet, or if you haven't been to lab meeting, then you may not realize that the red color doesn't just mean one thing, and that in this two-step test. So I'm just going to tell you the answer, and it hopefully is in your head later when you revisit it. But in this two-step test, if you turn red after the first step, then that's positive. And if you turn clear after the second step, that's also positive. And if you turn red after the second step, that's negative. So read about the nitrate test. Okay, exoenzyme testing is something that you have access to. This is an example. This is called a milk plate, but what it really tests is, can you hydrolyze casein, which is the opaque protein in milk? Which side of this plate has an enzyme that makes the exoenzyme caseinase? So the milk plate is literally got milk in it. That's why it's opaque. And what we can see is these two different species. This one makes an enzyme. So this part here is the streak. It's the growth of the bacterium. And this one makes an enzyme which is secreted outside, and it digests the casein, and so the milk clears. All right, here's another exoenzyme test. This is a lipase plate. So this blue dye is lipid-based dye, and an organism that has lipase will clear the dye. And I don't know why this is totally swapped. And I'm sorry, guys, that must be a little confusing. This is positive, that's negative, obviously. I have to fix that. I don't know how many times I've proofed these slides and I still see mistakes in them. Sorry about that. But the class of macromolecule being digested, of course, is the triglyceride. Here is another exoenzyme test. This is the starch plate. So the plate is made with starch in it. And then you grow your bug on it, and you can't see when it's first grown if it worked and if it digested the starch. So what you do is you add iodine. Iodine is a chemical that binds to starch and turns it dark. Which of these represents, the left or the right, represents the organism that can digest the starch? Yes, obviously the starch has been digested here. That's why the iodine didn't have anything to bind to. And we've already talked about the enzyme. It's the amylase test. So starch, you can call it the starch test or the amylase test. So you may see either of these wordings on your Bergey's table. All right, my plan was to do a Kahoot, but we only have 7 minutes, and I'm sort of wondering if you'd want to, and it might go a little long. So we could do the Kahoot at the start of class on Tuesday and just save these last few minutes for questions. Is that what you guys would prefer? Do you want Kahoot or do you want questions? Okay. Yeah, let's do the Kahoot on Tuesday, and that'll kind of help because then you'll have had the weekend to kind of digest these ideas, and the Kahoot will kind of be a like reboot and see if we remember and understand things. Man, my brain is at lunch too. I didn't even eat breakfast this morning, so I am ready to eat my computer. All right, so we'll do the Kahoot on Tuesday. I do want to take a second to just remind us where we are in our unknown ID process. I'll also talk about this in lab meeting, but we are coming up on the deadline for the group ID. So make sure that you're well on your way through the part 2 analysis. And it does take, you know, more than a day. So if you haven't started this yet, you are really behind. Get started now and just kick butt as fast as you can. When you refer to just the group, do you use italics? No, the group is large enough that even, so some of the groups are not even genuses, they're multiple genera, and they're definitely not italicized. But even a genus, when the genus is just mentioned by itself with no species name next to it, it's not usually italicized. You don't have to do that. Tonight, pre-lab 4 and discussion 4 are due. So don't forget to do that. And if you are already done your group ID, you can submit and move on to step 3. You should at least read step 3 and see if you can get finished in the next little while, but Sunday is the deadline. So Sunday, a reminder, metabolism review quiz, Dropbox for lab 4 and post lab 4, and then unknown ID part 2 group ID. Homework for Tuesday, basic, as if that wasn't enough, right? Oh my goodness. Basic genetics page, it has a little quiz that goes with it that's not for credit. And the bacterial genetics page, so watch the video lectures. Genetics is another heavy topic like metabolism. It's also lots of fun, but it's way more fun if you've done the prep and you come to class ready to sort of deepen your understanding. All right. I will see some of you in lab meeting. Feel free to sign out if you're done. And I will take questions now. Let me just stop my recording.